Hello, everyone, and welcome to today's webinar, Mastering Small Molecule Reversed Phase Method Development. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting as many questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you happen to have any trouble seeing or hearing the presentation. I would now like to welcome our speaker, Dr. Adham Ahmed, Global Product Manager, Thermo Fisher Scientific, UK. For a complete biography on our speaker, please visit the presenter window at the top of your screen. Dr. Ahmed, you may now begin your presentation.
Everyone to today's webinar on small molecule reverse phase method development. I'll be covering various topics to provide you with an overview of the key factors that can impact your chromatography, but also what you need to take into account when you're doing any method development. The first topic I'll be going over is how to get a good chromatography. One of the key goals in chromatography is understanding the factors that have the biggest impact on your chromatography and how to tune that method you're trying to develop. Some of the key factors in any chromatographic method development is to tune the retentivity of your sample, allowing sufficient time for the sample of interest to pull apart and achieve a good baseline resolution between two analytes. The next crucial factor is selectivity, which refers to the type of interaction occurring between the stationary phase and the analyte of interest, providing the resolution between the analyte. Finally, efficiency plays a significant role in the separation performance. The higher the efficiency of the column, the sharper and narrower peaks would be, indicating superior column performance. So here's a good illustration of a good and poor efficiency and selectivity between two analytes. In a good separation, the peaks are sharper and we achieve a good baseline resolution between the critical pairs. In a poor selectivity and efficiency, we get wider taily peaks and coeluting of the critical pairs. To reach that point, it's important to understand the key parameters that help in controlling each factor. In the next few slides, I'll go into more details on how to optimize each of these factors to give you an idea how each parameter can influence your overall result. Efficiency is one of the key parameters in chromatography. In the next few slides, I'll be going over certain parameters that would have an impact on the column efficiency and how we can improve the efficiency of our methods. How column efficiency is measured. Here we have the Van Meter equation, which is derived from three factors, A, B, and C, which contribute to the overall column efficiency. If we start with the first factor, and that is the A term, is known as the eddy diffusion, which is the route the molecule will take as it travels throughout the column, as shown here in this animation. This term is affected by the non-uniform flow of the molecules as they travel throughout the column. The A term is minimized by using a well-packed column with uniform particle size and size distribution. And that is one of the key parameters that we focus on when we manufacture our particles, is by having a tighter particle size distribution to improve the flow uniformity throughout the column with well-packed columns. Next, we have the B term, which describes how analyte diffuses throughout the column and is strongly affected by the mobile phase velocity. The lower the velocity of the mobile phase, the bigger the B term. As the molecule travels slowly through the column, there's a higher chance of band broadening, as seen here by this animation. So things like how well the column is packed and reducing the multi-channeling throughout the column would be the key factors in improving the B term. The last term is the C term, which is the mass transfer, and that is the rate of how molecule diffuses into and out of the pores of the particle, and also the interaction of the molecule with the stationary phase. This is a nice animation which shows how the rate of mass transfer is larger for bigger particles compared to a smaller particle or a core shell particle. This becomes more important as we go to higher mobile phase velocities, where the C term remain flatter for smaller particles, and this helps increase mass throughput with little loss in efficiency. We understand the importance of each of these terms in driving the column efficiency. Throughout our particle manufacturing, we maintain at tighter specifications, and we continuously monitor our column packing efficiencies to ensure optimum column performance. When we combine all three factors together, A, B, and C, we obtain the overall Ventimeter plot, which is a plot of plate height against linear velocity as shown on the left hand side of the slide. As we go smaller in particle size, the plate height is reduced, and that is because the column efficiency has increased. What's also important to notice is the C term. It becomes flatter as we go from bigger particles to smaller particles. So we are able to get efficient separations at higher linear velocity. One thing to notice is the pressure, which is inversely proportional to particle size. As the particle size decreases, the pressure increases. So it's important to have the right system for the column you're using to fully leverage the column performance and also improve sample throughput as well. So when you're developing any new method, it's important to consider the effect of column dimension and the choice of particle. So in terms of the choice of particle size, the type of the particle is quite important. As I mentioned in the previous slide, small particles improve the overall column efficiency and separation. The choice between totally porous and solid core, depending on whether you're aiming to push for high sample load or achieving a high efficiency with the solid core technology. In terms of the choice of column dimension, such as smaller column IDs can help reduce solvent consumption, but it's also suitable if you're actually dealing with very small sample. The column length, depending on your goals, shorter columns can reduce the runtime and increase the productivity of your lab, while using a longer column can be beneficial for separation of complex sample. There are definitely a lot of factors to consider when you're actually developing a new method, or even if you're actually modernizing your old method. Here we have our two particle platform, totally porous Hypsol gold column and solid core AccuCore column. Our Hypsol gold columns is our gold particle technology built on 40 years of experience in column development and manufacturing. It is fully porous, ultra pure silica particle, have been used and proven to provide excellent peak shape and lot of reproducibility for many years, and consistency you can rely on and result you can trust. It is available in a wide range of column sizes to accommodate your need, from narrow ID columns to a standard article size column, all the way to primitive scale 10 to 50 millimeter IDs for sample fractionation. If you are looking to resolve all the impurity without feeling the pressure, AccuCore columns would be your perfect partner for your application. Our solid core silica technology consists of a non-porous core and a porous shell to help in increasing the mass transfer and maximize the efficiency of your separation. It is available in a wide range of chemistries to help you deal with more challenging separation. In this section, I'll be going into the selectivity factor and how this can improve selectivity of your method. The goal in chromatography is resolving a mixture into individual compounds. And what's important is achieving the best resolution. Resolution is how well two individual compounds or peaks are separated from each other, and is calculated as the difference of retention time of two eluting peaks divided by the average width of the two peaks at the baseline. Here on the slide, we have the resolution equation, which consists of three factors: efficiency, selectivity, and retention characteristic. Efficiency is affected by the column packing efficiency, the particle size, and the particle size distribution. Then we have selectivity, which is controlled through the mobile phase composition and the stationary phase. We also have the retention factor, which measures how strongly an analyte is retained by the stationary phase and is dependent on the mobile phase composition, the column dimension, and the particle size. Here on this chart rate, you can see that although the peak capacity and efficiency do impact the resolution, selectivity continues to impact the resolution of the method long after efficiency and retention factor. Selectivity factor is a crucial parameter that would affect the separation, efficiency, and resolution, and it's strongly governed by the type of phase used in your application. So it's important to remember when you're developing a new HPLC method, it's best to explore other phases to see how different selectivity can improve your overall chromatography. To pick a suitable column to start your method development is always the difficult step when you're screening a new sample. So it's important to understand what type of interactions you would get from the sample of interest. The first step is to identify the functional group and the chemical difference between the compounds you are looking to separate. If you have a lot of analyte in the method, you can look at the main differences between the analyte, or if you already have a method, but there is one critical pair that you are not able to separate because they are too closely related, then you can focus more on the critical pair to understand the differences. So it is a little trickier. The more analyte you have, the more complex you get. Usually there are a few analytes that you are struggling with, and then you can focus more on those to find a suitable column that would help to resolve those two analytes. Have a better understanding of the analytes we're trying to separate. We need to look at the difference in functional group. That can be the length or the density of hydrocarbon. Does it have aromatic ring, steric differences or isomers, nitrogen-containing function group, oxygen-containing function group. This can give an idea about the behavior of the molecule you're trying to analyze. Here we have 4-hydroxylphenyl acetonamide, which is an active ingredient in paracetamol. We need to break down the molecule further to understand what type of interactions we would get. Things to consider, as I mentioned in the previous slide, the length of hydrocarbon determines the degree of hydrophobicity. In this case, we have a small chain hydrocarbon. Does it have aromatic rings? And that would generate polar aromatic interactions. In this case, it does, and we would expect to get pi-pi interactions if we use a phenol base. Are we able to get hydrogen capacity with the stationary phase? So in here we have amide and hydroxyl group, and these functional group can form these type of interactions. We can see that 4-hydroxylphenyl acetonamide molecule would be considered as a more polar molecule with some hydrophobic properties. If this molecule is shaded ripetized, would alter the property of this molecule, and we will be able to tell if it's becoming more polar or more hydrophobic, depending on what group has been privatized. So it's important to explore the molecule of interest, identify the type of column you would actually use to screen that particular sample. So in this example, we are looking at the analysis of mebendazole and its impurity. It is a relatively hydrophobic compound with a carbamate group, which makes it interact strongly with non-polar stationary phases like C18 column. It also contains polar groups such as amide, esters, and carbonyl. Using a hypoxyl C18 column, we've achieved a nice separation for the API and its impurities. If we looked at other phases such as polar advantage or phenyl-hexyl, we can start to see a change in rotation time and elution order, and also an improvement in the baseline resolution for certain impurities. The retention time significantly changes with polar stationary phases, as now we gain a higher hydrogen interaction capacity. So it clearly stresses the importance of, when you develop an HPLC method, is to understand the molecule of interest and also to explore what other phases that can be used to see how that selectivity would change and any improvement that can be achieved by alternative selectivity. In this section, I'll be taking a deep dive into different phases and to give you more insight, what is the difference between hydrophobic and polar and aromatic phases, and what features they can offer to help you in fine-tuning the selectivity of your separation. Let's start with hydrophobic stationary phases. These are commonly used in reverse phase chromatography. This type of stationary phase consists of a non-polar hydrophobic material which interact with the hydrophobic portion of your analyte, allowing for the separation based on hydrophobic partitioning on the stationary phase. These phases separate molecule based on the degree of hydrophobicity. The more hydrophobic the molecule is, the higher the retentivity on the column. The C18 phase is widely known as a general-purpose column, which is a balance of a non-polar retention for acid, bases, and neutrals. It's a good starting point to help understand the behavior of a particular sample and to determine if a C18 phase will provide the desired separation. C18 ligand is a long-chain hydrocarbon, and that's where the analyte would partition during separation. It's important to understand that not all the C18s are the same. They would behave differently based on the bonding method and reagent used during manufacturing. We could have mono, di, trifunctional octodicyl silanes, which represent the primary bonding. The surface is then end capped to reduce any secondary interactions that would affect your chromatography. The percentage carbon determines the degree of surface hydrophobicity of a particular column. The higher the carbon, the more hydrophobic the surface would be. For all of our chemistries, our bonding have been optimized to achieve dense coverage to minimize any unwanted secondary interactions. Each chemistry is tested under tight specification to evaluate interactions such as hydrophobicity, ion exchange properties, selectivity, hydrogen capacity, acids, bases, and neutrals. And this has helped us to maintain the consistency and reproducibility of all of our products for many years. Here's a good example, the application of fat-soluble vitamins on an AccuCore column. If we look at the fluoroanalate in particular, the difference between K2 and K1 and D2 and D3 is the presence of double bond, which would introduce polarity to the molecule, but we would also get less hydrogen bonding with the stationary phase and causing them to elute much earlier. What's interesting, the difference between D2 and D3 is a single double bond, and the column was able to distinguish between those two molecules very easily. There is also other hydrophobic phases with shorter chains, such as C8 or even shorter. As we move to a shorter chain ligand, the column becomes less hydrophobic, allowing the analyte to elute much earlier. This can be useful if you want to shorten the runtime of your analysis, or if you are actually dealing with very hydrophobic compounds that are strongly retained on the column and they're quite sticky. Reducing the column surface hydrophobicity can be very beneficial in these situations. Here is a nice example where we're trying to separate a mixture of di, tri, and tetra amines. We can clearly see the shift in retention time dropping from 12 minutes, 10 minutes by simply reducing that surface hydrophobicity. So we can actually fine-tune the retentivity of our analyte by actually changing the degree of hydrophobicity of our column. Not everything can be done using a standard hydrophobic column. The need for alternative selectivity is quite important, especially if you are struggling to retain polar analyte. We offer a number of different phases, including polar end cap, polar embedded, and polar end group. These type of phases provide unique properties and can withstand 100% aqueous mobile phase conditions, which enable us to start with less than 5% organic in the mobile phase. I'll be going through some of these examples in the next few slides to show you the benefit of having polar modified surface. So here we have a polar end cap C18 column. The primary stationary phase is a non-polar C18, and this would provide us with the standard C18 properties. The surface is then capped or modified with a polar group to introduce a polar surface, allowing for polar interactions with the analyte. Polar end capping can lead to better retention of polar compound, as the column's surface is more compatible with polar interactions. In this example, we're trying to resolve a mixture of calcium molecules, and these are considered to have both hydrophobic and polar characteristics. They have a hydrophobic core structure due to the aromatic ring, but they also have polar hydroxyl group, which contribute to their polarity and hydrogen interaction. The hydroxyl gold column can interact with the hydrophobic core of the molecule, but we can improve the polar interaction by adding a polar modified column. We can clearly see the change in retention time due to the change in selectivity, but we also notice an improved baseline resolution, as now we are able to utilize the hydrogen capacity interaction with the polar modified column. It is important when developing a new method is to understand the molecule of interest and to explore different selectivity to fine-tune the chromatography to get the best result for your sample. In this example, we have a polar embedded functionality within a C18 phase, which is a quite unique phase compared with other phases. From this figure, we can see the structure of the polar amide embedded C18 ligand. The C18 portion of the stationary phase provides hydrophobic interactions, attracting and retaining non-polar molecules. The amide group on the C18 phase allows for stronger hydrogen bonding interaction with polar analyte, providing a unique selectivity compared to conventional C18 phase. The amide group can also contribute to ion exchange interactions, particularly with charged analyte at different pH conditions. This stationary phase has been particularly useful for retaining highly polar molecules and provides excellent peak shape for polar bases such as amino acids. In this example, we can see the impurity profile of aspartic acid and glycine, where we can achieve a good retentivity for these amino acids with excellent peak shape. Another unique interaction is hydrophobic steric interaction. There are limitations when you are dealing with certain molecules that have varying degree of bulkiness as well as varying length of hydrocarbon chains. Using a standard C18 ligand, this application would be quite challenging. To address these particular challenges, we have developed a special phase that can provide the right tool for this particular application. So here we have a special hydrophobic column phase, which is known as C30 phase. Well, it is similar to a C18 chemistry in terms of linear structure, but it offers a unique interaction due to its bulkiness and the way it would fold on itself. The C30 phase can fold to produce unique interactions with bulky molecules that no other phase can do. So here's a good example where we're actually trying to analyze lipid nanoparticles. In this study, we're able to understand the formulation of the lipid nanoparticles and the ratios of each component. This particular application is not possible to achieve using a standard C18 column, as it lacks that geometrical separation properties that is needed for more challenging separations. Lastly, we have the phenol phases, which contain orthogonal selectivity to the standard hydrophobic chemistry. The aromatic functionality allows for pi-pi and dipole interactions, which can be useful for the separation of molecules contain aromatic groups and provide unique interactions than standard C18 phase. I'll go through the different phases that we currently have available to give you some idea what are the differences between those phases and how they can provide you with unique properties for your applications. Here we have three very distinct aromatic phases, PFP, phenyl hexyl, biphenyl phases. What's interesting about the aromatic phases is the distribution of the electron cloud. So in the case of PFP, it contains fluoroatom, and these would act as electron withdrawing, pulling the electrons away from the aromatic ring. This creates a negative charge outside the ring and a positive charge inside the ring, which improves the interaction with other aromatic isomers and halogenated aromatic. For the phenyl, hexyl, and the biphenyl, the electron cloud creates a permanent negative charge in the middle of the aromatic ring, and thus becomes even stronger when we have two phenyl rings very close to each other, like the biphenyl. Each of them would provide a different level of hydrophobic, steric, electrostatic, and hydrogen bonding interactions. We can see how these phenyl phases can provide you with different level of properties for your applications, and I'll go through some of these in the next slide. Some aromatic molecules can be challenging to separate on a C18 column, so phenol phases are excellent for separation of aromatic and moderately polar analyte, as we have the different level of selectivity. What's also interesting is the phenyl hexyl phase, and in this case we have a bimodal selectivity coming from the long chain C6 hydrocarbon and also the phenol phase. So here we have a nice example where we're trying to separate derivatized dinitrobenzenides, and it can be observed that the three phase-eluting compounds vary by an amine or a methyl group. We can draw on the benefit of the aromatic interactions, and we can achieve a good baseline separation between these three molecules. Next, we have a nice example for the separation of steroid on both a C18 and a biphenyl column. These are a group of compounds that have one or more aromatic rings. Most of these molecules can be retained on a C18 column due to the volume of hydrocarbon present in the molecule. However, if we run this type of compound on a biphenyl column, we get both C18 interaction properties with the additional aromatic pi-pi interaction, increasing our retention time for this molecule, but also we see an increase in resolution of this compound. We can clearly see the benefit of adding a orthogonal selectivity when you actually do a method development for aromatic molecules. Lastly, we have PFP columns. Here we have an interesting application which demonstrates the unique selectivity of PFP column in the separation of a mixture of toluene and aniline compound. Peak 2 and 6, which are structurally similar and have a similar number of carbon atoms, would typically have a poor resolution on a CAT column. However, when we actually use a PFP column, these two peaks are well resolved, and that's because of the additional interaction that the PFP phase would provide. This highlights the superior selectivity of the PFP column in the separation of compounds that may coalesce in other stationary phases. In this last section, I'll be looking into the mobile phase conditions in method development. As we've already seen, looking at different phases is important in optimizing the separation. To drive that separation further, the mobile phase conditions is also an important part to consider, as we can control the retentivity and also the selectivity of our molecule, and would allow us to optimize our methods further. The simplest method in chromatography is isocratic separation, and that's because the mobile phase composition remained constant throughout the analysis of our sample. However, switching from isocratic to gradient method offers advantages in separating complex mixtures and can improve the resolution and also increase the analysis speed and sharpen the peak shape of our analyte. Gradient method provides better control over the separation of compound depending on the strength of the organic mobile phase and also the steepness of the gradient. The quickest and the most traditional gradient method involves running the gradient from 5% to 95% organic over the length of the column. For example, if the column is a 10 centimeter, a 10-minute gradient profile would be a good starting point. It is important to remember to go to higher organic levels at the end of the gradient, and that's to wash and recondition the phase before the next injection. After you have identified a suitable starting point for your gradient method, you can adjust the gradient method further with steps to improve the overall method. If you're struggling with retaining early eluting peaks, one approach is to start lower organic composition to improve the retention of early eluting peaks. If you're planning to go below 5% organic at the start of your gradient, you would need to use a different column that can handle 100% aqueous mobile phase conditions. If you want to start moving the peaks around in your chromatography, you can do that by adjusting the steepness of the gradient or adding an isocratic hold over critical pair to see if this would allow for better separation or resolution for those peaks. In this example here, the gradient was modified to pull the last 3 peaks to shorten the run time. This was done by simply adjusting the steepness of the gradient over those 3 peaks, and we were able to pull that retention time from 10 minutes to 8 minutes, while still maintaining the resolution between those 3 peaks, and also we improved peak shape as well. So you can clearly see that when you're actually doing any method development, exploring different parameters within your mobile phase conditions and also the gradient conditions can have a big impact on your retention time and also the resolution of your analyte. The most common solvent in reverse phase chromatography are acetonitrile and methanol. Acetonitrile is a polar aprotic solvent, which means it can donate protons. But in the case of methanol, it can donate protons and can form hydrogen interactions. They behave differently and interact differently with the stationary phase and the analyte. A good example of how these solvents interact with the stationary phase is an aromatic phase. Methanol improved the interaction between the phenol stationary phase and the analyte, where in the case of acetonitrile, this type of interaction is suppressed. Methanol is considered as a weaker solvent, as the retention time would significantly be affected. One thing to remember between these solvents is the viscosity. Methanol has a higher viscosity than acetonitrile, and this means methanol water mobile phase create more pressure compared to acetonitrile mobile phase. So here's a nice example to see what happens when we swap different solvents. We can see the changes in chromatography when we move from acetonitrile to methanol. Because we changed the mobile phase, we're now altering the interaction that is happening inside the column. So here in this application, we're using hydrogol to carry out the QC test for pesticide standard. When we're moving from acetonitrile to methanol, we can see that the co-eluting peaks 6 and 15, as well as 14, 16, 8, and 9, are now resolved. And that's most likely due to the protic abilities of the mobile phase. The OH group has more active interaction with these analytes, and this helps us to achieve this increase in resolution for these analytes. We also see an increase in retention time of all the peaks due to methanol being a weaker solvent than acetonitrile. Here we're focusing on the stationary phase behavior with an organic solvent. In this case, we are using phenyl hexyl column. We clearly see the change in behavior when we swap him from acetonitrile to methanol. The retention time increase when we're using methanol in the mobile phase, and the resolution between peak 6 and 7 improved. What's also interesting is the elution order for peak 8 and 9. They have swapped, as now we're seeing an increase in interaction with the stationary phase. As we have already seen that methanol enhances that phenyl ring interaction with the analyte and give us different selectivity. So it's important when we're actually doing method development is to explore different organic solvent, as it does alter that selectivity of the stationary phase and help us to fine-tune our methods. As we have already seen so far, there are a number of parameters that needs to be taken into account when we're doing method development, from exploring different phases, column dimensions, type of particle and particle size, the method, whether it's isocratic or gradient, including the choice of mobile phase solvent. What's also important when you're doing a method development is exploring the total workflow. We have already seen how the column and the mobile phase condition can have an effect on our chromatography, but the rest of the workflow still contribute to the overall result of our analysis. When developing a new method, it's important to explore the full workflow to maximize the efficiency of your result. Things like the vials, which hold your sample, how clean is your sample that you're using during your analysis. These are key factors that control the quality and the reusability of your result. I would like to highlight that we offer comprehensive workflow solutions covering everything from vials and sample prep to columns and instrument. We have developed several workflows for key applications such as nitrosamines, PFAS, and many interesting workflows as well. If you are interested in exploring these further, you can find out more information on our website. One thing that a lot of HPLC users don't take into account is choosing the correct vial. Vials are the most important starting point as they hold a sample of interest. Ensuring that you're actually using the right vial for your sample is critical. Our show of start vials are available in 3 performance levels, ranging from everyday analysis to high-performance selective and sensitive vials. Choosing the right vial ensure reliable and reproducible data and confidence in your results. Another point to consider in your workflow is sample preparation. There are several reasons why sample preparation is performed on a sample prior to analysis. One of the main reasons is how clean is your sample of interest. In this case, sample preparation helps remove the analyte of interest from other components of the material. The matrix of your sample can significantly affect the result in several ways. It might collude with the analyte of interest, leading to overlapping peaks, can interact with the stationary phase, altering the retention time of your analyte, can affect the detection response to the analyte, and in the case of mass spec, it can cause ion suppression. So there's many factors that the matrix can have a big impact on your overall chromatography. Another important reason is system robustness. Removing the matrix prevents column and system from getting dirty or contaminated. This would improve column and instrument lifetime and reduce unnecessary costly maintenance. Thank you all for your time and for joining us today. I'll be happy to take any questions you may have.
All right. Perfect. Thank you very much, Dr. Ahmed, for your informative presentation. We will now move into the live Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We will answer as many of your questions as we have time for. Okay, let's take a look at the first question we have here. This one says, how do you ensure that a newly developed method is both robust and reproducible?
Thank you. I mean, that's quite an interesting question. I mean, the way I see it is robustness and reproducibility go hand in hand. So, I mean, that can be done by the analysis of the same sample and the various conditions determine the ability of the method to stand the variability conditions. And what I mean by that is, I mean, that can be done by actually intentionally introducing variability into the actual method to see how that particular method will behave and how that variability can stress your actual method that you're developing to see how reproducible it is. Variability in the condition normally is expected from lab to lab, and that's quite normal. Going from lab to lab, there's variability, instrument to instrument, and also from analyst to analyst. So it's important to build that into the method to understand the reproducibility and the robustness of the method without significantly affecting its performance. So things to take into account, for example, the mobile phase composition, the ratio of the organic solvent, how the variability of that, examining that to see how the method vary from one operator to another, from one preparation to another. That gives you an indication of the variability that you'll be getting from the mobile phase by itself. Things also to take into account, like the buffer composition, the aging of the mobile phase is how, when you leave the mobile phase on the system, how long does it take to age? And that will also introduce additional variability, and it gives you an understanding how long you should actually refresh that mobile phase before you introduce that effect into your method. The pH of the mobile phase, how does that change over time? Also another factor. What's also important is different columns from different lots, and that is quite crucial in order to help you understand the variability that is coming from the column, to build that into your method, to make sure that the method would actually be able to consider all the variability that's coming, not only just from mobile phase, but also from the column chemistry, and also the temperature of the lab. That's also another variability. So there's a lot of factors that need to be taken into account. And well, also also interesting to consider is the sample stability and sample degradation. So there's various factors that can be looked into to assess the robustness of that method and make sure that the method is quite robust and reproducible.
Perfect. Thank you so much. Okay, it looks like we have another question here. This one says, what type of columns are most appropriate for low molecular weight hydrocarbons?
I mean, type of column, I mean, usually just any column that would be standardized for small molecule separation. I mean, these are quite small molecules, so anything with the pore size between 10 nanometer to 120 nanometers should be sufficient and should be able to withstand that. Again, it's important to understand the molecule you're trying to separate, as we explained that in the slides, to understand what type of interactions you're getting, understand what type of are you getting mainly hydrophobic interactions that you begin from that particular molecule. Are you going to get any polar interactions, and also if there's any phenols present in the molecule, and that could give you an idea what type of stationary phase to select in order to examine and explore, in order to see which chemistry gives you the best separation for that molecule of interest.
All right, fantastic. Next question here says, what are some common challenges faced during method development and how can they be avoided?
One of the main challenges in understanding, one of the main challenges in method development is, as I just mentioned, is understanding that molecule. First of all, it's important to define the interactions that you'll be getting from that molecule of interest, so in order to determine the specific interactions you begin with that stationary phase, and then things to consider is how that can help you to fine tune the selectivity and also the resolution for that critical pair. For example, if you are struggling to separate or to retain ill-eluting compound, then you would actually try to consider things such as moving to polar phases in order to help you retain these molecules, or changing the gradient. It depends what factors is driving that separation in the molecule. So another point is to look at the full weight flow of your method, and this is one of the challenges as well, is to make sure that we get in reproducible delivery of the sample. If the sample in the matrix and we're not actually removing the matrix properly or effectively, then we're actually introducing challenges into our method and would have a big impact on our retention time, reducing the detection of or the sensitivity of our detection, and also could have a big impact and challenges to the life of the column. Another would be is to stress the method, and I think this is what I just mentioned previously with the first question, is to to build in that stress in that method. To obviously, once you develop that method, is you need to make sure that all variability is taken into account to build and to help build that method even more robust. And this will help reduce some of the challenges faced when you're actually moving from column to column, or changing that mobile phase from an older mobile phase to a new mobile phase, or from one system to another.
All right, great. Thank you. Looks like we have a few more here. This next one says, how do you select the right HPLC column for a new sample?
That's quite a good question, and that does follow up from question number 2. Identifying the type of analyte you're trying to analyze, that's quite important, because that would be the starting point to help you determine whether you actually go into reverse phase or helic or ion exchange chromatography. So choosing the right selectivity or stationary phase is the most important part with choosing the right column, and that's by understanding the molecule that you're trying to separate. Identifying functional groups, the chemical differences between the compound. If you have a mixture of very complex sample, then things to consider is what would be the main differences between those analytes to help you identify the next step. Another point would be considering the column dimension and the particle size. Obviously, if you are considering, if you're looking at reducing solvent consumption, then you go into a small ID columns to help with that particular goal. Or if you are working with small amounts of samples, again, smaller ID columns would be an ideal option to consider or to choose for a new sample. The column length, depending whether you're actually trying to achieve a shorter runtime, or if you're dealing with a very complex sample, then you need a longer column in order to help with increase that efficiency and retain the molecules longer to help resolve such a complex sample. Again, what's also important, and I stress this as well in the slides, is the particle size and the type of particle. Again, things from a particle perspective is the surface area, the pore size, depending on the analyte you try and separate, the choice between totally porous or solid core particle, depending whether you're actually trying to push for higher sample load or trying to achieve a high efficiency with a solid core technology, and that would depend. That would depend what you're trying to achieve from your method, and also the choice of the column as well. So there's a lot of factors that need to be considered when you're actually selecting a suitable column for the method. And but the first step, I would say, is always looking at the sample of interest, and then try to explore what type of interaction you'll be getting and what type of columns you'll be looking to explore.
All right, perfect. We have another here that says, what are the effects of pH on the separation?
pH, that's a good question. Again, this is one of the points we did not really touch on in these slides. Obviously, the pH is quite an important factor. It influences the ionization of the analyte of interest, which really affect the retention time and also the interactions with the stationary phase. And that's quite important, especially if you're actually working with acids and bases, where these can actually be protonated, deprotonated, depending on what pH you're actually using. It wouldn't really affect neutral molecules because theirs will not be affected at the pH, what pH you're using for those. So in most cases, when you use an acid or bases, the mobile phase must be 2 pH units below the pK of the molecule to obtain a fully protonated species. And then if we're looking at acids, for example, at lower pH, these would become more protonated and would actually see an increase in retention time. So we can clearly see that moving to a lower pH, we actually increase the retention time for acids. The opposite will happen for bases as these becomes more ionized, and then we actually start losing that retentivity for those bases. So it's important to consider when you're actually doing method development is the pH of the mobile phase, and also whether you have acids and bases in order to balance or find the right pH range for your analysis.
All right, fantastic. It looks like we have one final question here. And this one says, what are the best practices for prolonging column life in HPLC?
Yeah, I mean, this is quite an important question, and again, that would really depends on, and it's basically, I would say I would start off with basically understanding the column you're using and its limitation, making sure that you're operating the column at the recommended conditions, including the pH, the temperature, and the pressure limitation of it. the storage condition of that column. All these will have a big impact on the column lifetime. I mean, this is the first step. What's also important is sample cleanup, which I touched up on that on the last part of the presentation. Sample cleanup, filtering the sample to remove any particulate, removing the matrix from the sample of interest. All these will have an effect on the lifetime of the column, and also the performance, and also the variability of the data. So sample preparation, sample filtration would have a big impact on column lifetime. You can use guards. Guards can be used to prevent contamination from reaching the column. Also, routine column washes reduce the risk of column contamination and the column being clogged. Also, if you're actually using buffer to wash the buffer from the column. So there's a number of things that need to be considered when you actually using a particular method, is to see what factors will have the big impact on the lifetime of the column. As I mentioned, the buffer, what type of matrix is in, is the sample filtered? Are you using guard? Guard will definitely extend the life of the column, and also the storage condition of the column and the conditions. So we have a lot of HPLC column care guide and method development guides as well available to provide more details around that topic. And also the customers can reach out to our tech support team if they have any specific questions or about a particular column, and they'll be happy to provide them with additional details.
All right, perfect. Thank you very much, Dr. Ahmed. I did want to address one question that we received regarding the certificate of attendance, and that is downloadable via the event website. So underneath what will now be the red watch on demand or register button, there is a 12-pointed star that can be clicked on in order to download your certificate of attendance. So that is where attendees can find those. With that, thank you again, Dr. Ahmed, for your time today and for your very important research. We would also like to thank our sponsor, Thermo Fisher Scientific, for underwriting today's webcast. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact info that you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.